Hi, my name is Nur Sofia bint Abdul Hafiz, and today my group, which is Group 2, will be presenting about novel coronavirus. Our group member consists of 4 people, which is me, Nur Sofia bint Abdul Hafiz, Leong Shin Lek, Muhammad Alif Shazwan bin Muhammad Azizan, and Hasanah Fikri bin Abdul Ghani. So for our main topic is novel coronavirus, or as known as SARS-CoV-2 or Severe Acute Respiratory System CoV-2. The session number of NC045512 and the genome size of 29903 BP. So this is the model for SARS-CoV-2 and it consists of 4 main protein, which is E protein, S protein, M protein, and HE protein. E protein is a small protein in the coronavirus, which it is a small member protein that has an important role in the assembly of virions. Virion is actually an entire virus particle, which it consists of an outer protein shell called as capsid and an inner core of nucleic acid, either ribonucleic or deoxyribonucleic. For S protein, it is the main protein in all of the other proteins. From the M protein, E protein, HE protein, S protein is the main protein for coronavirus or SARS-CoV-2. It is actually responsible for inducing the host immune response, and not targeting the S protein can induce protective immunity against viral infection. This is the M protein. So the M protein of coronavirus actually plays the central role in a virus assembly. It can turn the cellular membranes into workshops for virus, and host factors come together to make new virus particles. Lastly, for the HE protein, or we call them as hemagglutinin esterase, it's actually an envelope protein and it serves as a receptor destroying enzyme.
Assalamualaikum and hi, my name is Alif Shazwan. I will show you about our original research paper that we have found about SARS-CoV-2, which is coronavirus. This is our article front page about coronavirus original research paper. Basically, this research is occur in Malaysia, and this is the reference for the citation of our article. This article is show about research on phylogenetic tree of SARS-CoV-2. They want to find the different species of coronavirus that have been pandemic in world. So several sample has been collect, which is from Malaysia, of course, Japan, China, Singapore, and US. So this the different sample get collect to different their characteristic from each clade. Basically, in this article, it show that it used 2 bioinformatics tools, which is cluster omega and molecular evolutionary genetics analysis, or we call it MEGA version 7.0. The cluster omega is to compare the strain from the sample and differentiate their percentage in index matrix. On the other hand, MEGA version 7.0 will create a phylogenetic tree result as in the proper shape of phylogenetic tree. Both of these bioinformatics tools is used in this research. So next, this is the result after they use the mega, it show that it create a proper phylogenetic tree and show different clade from different species, and this is the result of single nucleotide variants. Based on the name, only single nucleotide have have a variance in the strain. So the blue box show that it is a synonymous mutation, which is the product will be a same protein but only a different nucleotide. On the other hand, this is the amino acid nucleotide variants, it will show that the yellow box have a different protein that will produce from other strand. Okay, that's all from me. Thank you.
Hi, Dr. Sue and fellow friends. So as we know, Cluster Omega is a series of widely used computer programs used in bioinformatics for multiple sequence alignment. So it has pros and cons. In the meantime, in size, it can deal with very large numbers, basically positive advantages, many tens of thousands of DNA or RNA or protein sequence, single or double-stranded, due to its use the embed algorithm for calculating guide traits. It performing fast and accurate multiple sequence alignment (MSA) of potentially large number of protein, DNA or RNA sequence, very accurate profile-to-profile alignment. However, the disadvantages is this algorithm requires a certain amount of memory. If the amount of memory is not available, depends on your computer, then Cluster Omega switches to Viterbi algorithm. Usually it uses forward algorithm, which is search in context of a hidden Markov model (HMM). It is used to calculate a belief state, which is known as filtering probability of a state of a certain time. So if it's switched to Viterbi, it is not very good. Then forward algorithm. So that is the disadvantage. So the second one is input and output. The advantages is that if it's storing an embed matrix of n 1/4 of 10,000 sequences, takes up to approximately 14 megapixel of memory. A full distance matrix would take up almost 400 megabytes. So it is suitable for modern desktop computers, especially for us students. However, if the n 1/4 of 100,000 embed matrix that take up to 220 megabytes, which the full distance matrix will require about 40 gigabyte. So we need a better higher end machine. So it is not suitable for a student, and it will take a very long time to take the full distance matrix. The number of sequence affects the resource requirements of the profile-to-profile alignment stage only indirectly, in that it influences how much the length of the intermediary profiles grow from the length of the individual sequence. This growth is difficult to predict and depends, amongst other factors, on the similarity of the sequence. So, the increasing of number of sequence will increase the number of profile-to-profile alignments linearly, and therefore the alignment will also grow in a linear fashion. Next is in misalignment. So, individual sequence and small profiles are most vulnerable to misalignment. On the other hand, why so? Smaller sequence are most vulnerable to misalignment, but larger profiles build more pseudo counts and more likely to resemble the final alignment. What does that mean? further count with bigger number makes a lot of probability. So hidden Markov model cluster omega only accept one external profile HMM if the unaligned sequence extend over multiple domains, and HMM only known for individual alignments, so the only one of these HMM can only be submitted. So an alignment sequence are arranged in such a way that all the sequence maybe have the same length. They will be arranged in the same length, but not actually the same as the short sequence were placed before. It was arranged longer to suit other sequences. So this is interpreted by a cluster omega as a valid alignment, while it is in fact is not. So the lastly is the command line driven. The current implementation of cluster omega is command line driven, so there is yet no GUI and no interactive menu. So the help file also contains many examples. We need to drag the help files by pressing cross and H for the help file to pop up, and elucidating the use of all individual command line arguments and the range of typical combinations of command line argument. So it is not very family friendly usage. That's all for me.
Hi, I'm Liang Xingde, and now I want to present about the closely related organism of novel coronavirus, that is bad RATG dirty coronavirus. According to research of Roman 96 and Aries COVID, phylogenetically SARS-CoV-2 is closely related 96 identity to rhinovirus and if a host used bad virus strain RATG dirty. Besides, according to Ronsians and their partner researchers, they used newly sequenced SARS-CoV-2 strains and 20 closely related coronavirus strains to do comprehensive genome research to determine the evolution mechanism of SARS-CoV-2 genomes, and they find that SARS-CoV-2 strains and RATG30 strains are closely related. To verify that, the cluster omega sequence elements tools have used to compare the DNA sequence of novel coronavirus and bad RATG30 coronavirus. Open the pairwise sequence elements, and for this time we choose the selections to run our result, and in the page we choose protein to DNA, and next go to NCBI for the coronavirus 2 and go to FASTA to copy all the elements. And now open the notepad and paste the elements to the notepad. For the coronavirus RATG30, also do the same things. Go to NCBI and FASTA. Open notepad. Copy and paste all the elements of the FASTA. And now go to the page structures and copy the elements of coronavirus 2 to the frame. And for the coronavirus RATG30, also do the same thing, copy all the elements from the notepad and paste to another frame. double confirm the organism is right, and now we can submit and waiting for the result. And now the result is come out, and we can see the identity and also the similarity of two organisms have 96% of similarity. And that's all for the part of the closely related organism of novel coronavirus. Thank you.
Okay, so this is the analytical method of this research. Basically, they gather several samples from NCBI and GISAID. Then the sample is taken from a patients from Malaysia, China, Japan, and US. After that, they use cluster omega to compare the nucleotide and see the index matrix of the differences of the nucleotide. Then they check the phylogenetic tree result in the cluster omega result. After that, they use mega to create a proper phylogenetic tree of the sample. So they will give a better result for the phylogenetic tree. And lastly, they compare the single nucleotide variant and amino acid variant. Okay, that's all from me. Thank you.